Chapter 2, Phylogenetic Systematics. Overview and Goals. As introduced in the previous chapter, phylogeny refers to the evolutionary history or pattern of descent of a group of organisms and is one of the primary goals of systematics. Phylogenetic systematics or cladistics is the branch of systematics concerned with inferring phylogeny. Ever since Darwin laid down the fundamental principle of evolutionary theory, one of the major goals of the biological sciences has been the determination of life's history of descent. The phylogeny of organism, visualized as a branching pattern, can be determined by an analysis of characteristics from living or fossil organisms, utilizing phylogenetic principles and methodology. As reviewed in Chapter 1, a phylogeny is commonly represented in the form of a cladogram, or phylogenetic tree, a branching diagram that conceptually represents the best estimate of phylogeny. The lines of cladograms are known as lineages or clades. Lineages represents the sequence of ancestral descendants population through time, ultimately denoting descents. Thus, as previously reviewed, cladograms have an implied but relative time scale. Any branching of the cladogram represents lineage divergence or diversification, the formation of two separate lineages from one common ancestor. The point of divergence of one clade into two is termed a node. The region between two nodes is called internode. Figure 2.1. Example of a cladogram or phylogenetic tree for taxa A to F, with amorphous indicated by thick hash marks redrawn from Chapter 1. See text for explanation of terms. Figure 2.2. Cladogram of Figure 2.1 drawn horizontally with branch length scaled to number of amorphic changes. Evolution may occur within lineages over time and is recognized as a change from pre-existing ancestral, also called plesiomorphic or primitive, condition to a new derived, also called amorphic or advanced condition. The derived condition or apomorphy represent an evolutionary novelty. As seen in Figure 2.1, an apomorphy that unites two or more lineages is known as synapomorphy. One that occurs within a single lineage is called the automorphy. However, either may be referred to simply as an apomorphy, a convention used throughout this book. Cladograms may be represented in different ways. Figure 2.2 shows the same cladogram as in Figure 2.1, but shifted 90 degrees clockwise with the lineages drawn perpendicular to one another and of length reflective of the number of apomorphic changes. Why study phylogeny? Knowing the pattern of descent in the forms of cladogram can be viewed as an important end in itself. The branching pattern derived from a phylogenetic analysis may be used to infer the collective evolutionary changes that have occurred in an ancestral descent population through time. Thus, a knowledge of phylogenetic relationships may be valuable in understanding structural evolution as well as in gaining insight into the possible functional adaptive significance of hypothesized evolutionary changes. The cladogram can also be used to classify life in a way that directly reflects evolutionary history. Changes, cladistic analysis may also serve as a tool for inferring biogeographic and ecological history, assessing evolutionary processes, and making decisions in conservation of threatened or endangered species. The principles, methodology, and application of phylogenetic analysis are described in the remainder of this chapter. Character analysis description. Fundamental in any systematic study is description, the characterization of the attribute of features of taxa using any number of types of evidence. A systematist may make original description of a group of taxa or rely partly or entirely on previous published research data. In any case, it cannot be overemphasized that the ultimate validity of a phylogenetic study depends on the descriptive accuracy and completeness of the primary investigator. Thorough research and comprehensive familiarity with the literature on the taxa and characters of concern are prerequisite to a phylogenetic study. Character analysis. Character selection and definition. After taxa are selected and the basic research and literature survey are completed, the next step in a phylogenetic study is the actual selection and definition of character and character states from the descriptive data. Generally, those features that are genetically determined and heritable, termed intrinsic, are relatively invariable within OTU and denote clear discontinuities from other similar characters and character states should be utilized. However, the selection of a finite number of characters from the virtually infinite number that could be used adds an element of subjectivity to the study. Thus, it is important to realize that any analysis is inherently biased simply by which characters are selected and how the characters and character states are defined. In some cases, certain characters may be weighted over others. See later discussion. Because morphological features are generally the manifestation of numerous intercoordinated genes, and because evolution occurs by a change in one or more those of genes, the precise definition of a feature in terms of characters and character states may be problematic. A structure may be defined broadly as a whole entity and several components. Alternatively, discrete features of a structure may be defined individually as separate characters and character states. For example, in comparing the evolution of fruit morphology within some study groups, the character fruit type might be designated as two character states. Berry versus capsule, or the characteristic of fruit may be subdivided into a host of characters with their corresponding states. For example, fruit shape, fruit wall texture, fruit dehiscence, and seed number. These characters may be correlated, however, see later discussion. In practice, characters are divided only enough to communicate differences between two or more taxa. However, this type of terminological attenuation may be misleading with reference to the effect of specific genetic change in evolution. As genes does not normally correspond one for one with taxonomic characters, the morphology of the structure is the end product of development, involving a host of complex interaction of the entire genotype. Character state discreteness. Because of phylogenetic systematics entails the recognition of an evolutionary transformation from one state to another, an important requirement of character analysis is that character states be discrete or discontinuous from one another. Molecular characters and their states are usually discrete. See Chapter 14. For some non-molecular qualitative characters such as corolla color, the discontinuity of states is clear. Example, the corolla is yellow in some taxa and red in others. But for other features, character states may not actually be clearly distinguishable from one another. This lack of discontinuity often limits the number of available characters and is often the result of variation of a feature